Next presentation is implanted HAP stem cells encapsulated in polyvinylidenefluoride membrane cylinder promote effective recovery of peripheral nerve injury. Dr. Koya Obara. If you prepare your presentation, please begin.
My name is Koya Obara. Title is Implanted Hair Follicle Associated Pluripotent HAP Stem Cell encapsulated in polyvinylidenefluoride membrane cylinders promote effective recovery of peripheral nerve injury. This is the vocation. This is diagram of hair follicle. There are two types of stem cell. Missing expressing HAP stem cell and creatine 15 expressing stem cell. HAP stem cells can differentiate cardiac muscle cells, keratinocytes, melanocytes, smooth muscle cells, neurons, and glial cells. Purpose of this study is encapsulated HAP stem cell in polyvinylidenefluoride membrane cylinders, PFMC, promote effective recovery of peripheral nerve injury. Method: isolation and culture of GFP HAP stem cells and transfer to PFMC. Isolation of Verista hair follicles from GFP mice. Next, culture of GFP HAP stem cells. Then, transfer to PFMC. Implantation of GFP HAP stem cell PFMC to cerebrospinal nerve. GFP HAP stem cell PFMC was implanted the cerebrospinal nerve. Implanted GFP HAP stem cell PFMC rejoins cerebrospinal nerve. This is PFMC. This is rejoins cerebrospinal nerve. And GFP HAP stem cells grow in the cerebrospinal nerve. Working analysis of mouse after autologous implantation. Implantation of empty PFMC. The mouse cannot use right hind leg. On the other hand, implantation of HAP stem cell PFMC, the mouse is working normally. Conclusions. We demonstrate the implantation of HAP stem cell PFMC promotes effective recovery of the severed sciatic nerve. Thank you for your attention.
Thank you for your presentation. Do you have any question?
Ratapon from Bangkok, Thailand. Do you have any histology of the nerve after it regrow again?
You have your backup slide. Well,
This is implanted HAP stem cell PFMC injured sciatic nerve. Many spindle cells join in several sciatic nerve.
does that answer your question?
What is the
Can you come to the microphone, please?
I would like to know the difference between the control site and the transplant with HAP stem cell nerve. The difference between control and treatment.
The control did not rejoin. So Dr. Obara doesn't have that slide, but I can tell you that it was similar to the control I showed, no rejoining. There's no spontaneous rejoining. I think that's a key point.
Thank you.
And you can see he got rejoining very beautifully.
Excellent.
You're welcome. Thanks for a nice question. Any more questions? I'd like to make a small comment. This was Dr. Obara's first presentation in English, and I think you did a great job.
Thank you for your presentation. Next. Next presentation is functional validation of candidate Typogens genes identified from 3D cultured human dermal papilla cells by Yang K. Sung. Dr. Sung, please start your presentation.
Thank you for the introduction. I'd like to thank organizing committee for oral presentation opportunity. Today I'm going to talk about functional validation of some candidate typogens which we identified from 3D culture BP cells. As you know, dermal papilla is a mesenchyme-derived cell aggregate and regulates hair follicle development and growth. Many studies showed that adult fresh dermal papilla retains trichogenicity, and early passage cultured murine dermal papilla cells retain the ability to direct epithelial cells to form hair follicles. However, these cells lose trichogenicity during subculture. In case of human dermal papilla cells, trichogenicity is lost as early as passage 1. As shown here, trichogenicity of fresh dermal papilla is full, whereas trichogenicity of 2D cultured dermal papilla cells, trichogenicity is empty. To restore the trichogenicity of 2D cultured dermal papilla cells, two major strategies were employed. One is supplementation of activators of dermal papilla signature pathway, such as Wnt38, Wnt10B, Bio, BMPs, and FGF2. The other strategy is three-dimensional cultivation of dermopapilla cells. But 10 years ago, Osada et al. Made mouse vibrissae dermopapilla spheres using raw cell binding plates. And they combined these spheres with newborn mouse epidermal cells and injected into the nude mouse. And they found that these spheres retain hair inductivity. 5 years ago, we made human scalp dermopapilla spheres from dermopapilla from alopecia patient using hydrocell plates. And as shown here, spheres, human DP spheres induced hair follicles, whereas 2D cultured cells did not induce any hair follicles. More recently, Higgins et al. Made human scalp dermopapilla spheres using hanging graph method, and they placed these spheres between the epidermis and dermis of neonatal foreskin and grafted these recombined skin onto the back of skin mice. And they found, after several weeks, they found these human scalp DP spheres induce hair follicles. We questioned why there is a restoration in typogenicity when 2D cultured DP cells are prompted to form spherical structure, and we assumed that some tricogenic genes are upregulated in DP spheres. To identify these candidate trichogenes of human dermopapilla cells, we compared gene expression and secretory proteins between the non-trichogenic 2D cells and trichogenic DP spheres. Microarray analysis showed that a number of genes are differentially regulated, and genes such as SFRP2 and alkaline phosphatase is upregulated in DP spheres. Also, proteome profile array analysis showed that some secretory proteins are differentially regulated in DP spheres, and proteins such as activin A are upregulated in 3D DP cells. For the functional validation of candidate trichogenes, we adopted siRNA-mediated gene knockdown in DP spheres and in vivo patch hair reconstitution assay. As shown here, control siRNA or trichogen-specific siRNAs were transfected into the 2D cultured human dermopapilla cells for 48 hours. And following sphere formation for 48 hours, we combined these spheres with newborn epidermal cells and injected into the nude mice for 2 weeks. As shown here, SFRP2, in the case of SFRP2, knockdown by siRNA was confirmed by immunostaining of SFRP2. And as shown here, SFRP2 knockdown in DP spheres significantly impaired hair follicle induction. To confirm correlation between the trichogenicity of DP spheres with SFRP2 levels, we made DP spheres with dermopapilla cells from frontal balding, occipital, and beard DP cells. And we found that, as expected, we found the sphere formation increased the SFRP2 levels by sphere formation. And when we performed patch assay, we found that beer DP spheres that has the highest SFRP2 level induced was most trichogenic. So this data clearly shows the trichogenicity of DP sphere is correlated with SFRP2 level. Next, we investigated how SFRP2 increases trichogenicity of DP cells. And Western blot analysis showed that Wnt38 mediated accumulation of nuclear beta-catenin was augmented by addition of recombinant SFRP2 protein. In addition, reporter assay showed that Wnt38 mediated elevation. Wnt38 mediated elevation of beta-catenin topless activity was significantly increased by transfection of SFRP2 plus expression plus mute transfection. So these data shows that SFRP2 augments Wnt beta-catenin signaling, which is a very important BP signature pathway in BP cells. Now I'm going to move on to alkaline phosphatase and hair induction. Alkaline phosphatase activity has been regarded as an indicator or marker of trichogenicity of dermal papilla cells and dermal sheath cup cells. And as shown here, alkaline phosphatase activity is lost in 2D cultured dermal papilla cells. However, ALP activity is restored by sphere formation as early as 24-hour sphere formation. Alkaline phosphatase knockdown by siRNA was confirmed by real-time PCR analysis and ALP activity staining. As shown here, ALP knockdown in human DP spheres significantly impaired hair follicle induction. Proteome profiler analysis showed that activin A is one of the most upregulated secretory proteins in trichogenic DP cells. And we confirmed that activin A expression is significantly increased by sphere formation by real-time PCR analysis and immunostaining. Activin A knockdown in human DP sphere, as shown here, activin A knockdown in human DP spheres impaired hair follicle induction. To confirm the importance of dermal papilla sphere-derived activin A, we performed patch assay with activin receptor 2B knockdown mouse epidermal cells in combination with the human DP spheres.
Excuse me, Dr. Sung, can you wrap up pretty soon?
Okay. Yeah.
Thank you.
Yeah. So as shown here, activin receptor 2B knockdown in epidermal cells significantly impaired hair follicle induction. This is the summary. The trichogenicity of dermal papilla cells is restored by 3D spheroid cultivation, and a number of genes are up and down regulated by sphere formation, and DP spheres with knockdown of SFRP2, alkaline phosphatase, and activin A are severely impaired in hair induction. And restoration of these trichogen cocktails needs to be investigated whether these cocktails increase trichogenicity of DP cells. I would like to thank my colleagues and former students at Gyeongbuk National University School of Medicine and KNU Hospital Hair Transplantation Center. Thank you for listening.
Thank you for your presentation. So sorry we don't have time. But if you have any questions, please discuss in coffee break. I will close this session. Thank you for your cooperation.